All right, live stream's going. Hello, everyone. Welcome to the virtual seminar series on Bayesian decision making and uncertainty. And thank you all for joining us. We're really excited to have you all here. My name is Natalie Mouse. I'm a PhD student at the University of Pennsylvania, and I'm one of the organizers who'll be hosting the event today. I'd also like to thank my fellow organizers, Renato, Z, and Alex, for all of their work in helping to put this seminar series together. Before we get started with today's talk, I'm going to make a few organizational announcements. First off, following the seminar today, we've got an awesome lineup of speakers for the coming weeks. So you can find the schedule of the upcoming series, our seminar series speakers on our website. Our website is gp-seminar-series.github.io. So you can join future seminars via Zoom, provided you register on the Google group to receive an email with a Zoom link for each seminar. You can also register for this Google group on our website. Or otherwise, you can always just join via the YouTube live stream, which doesn't require any registration. All seminars take place at 1600 UT or UTC, so that's Coordinated Universal Time. And if you're joining us on Zoom today, you can ask any questions by just typing them in the Zoom chat. And then the speaker can choose to pause and answer these questions during the talk, or just to hold the questions and answer them at the end, depending on the speaker's preference. Okay, so with all that said, it's my pleasure to introduce Professor Paul Jensen from the University of Michigan, who will give today's seminar titled Exploring Phenotypes and Genotypes with a Robot Scientist. Okay, Professor Jensen, the floor is yours, if you want to go ahead and share screen.
All right. Did that work?
Yeah, looks great.
Okay. Well, thank you for the invitation to come and talk. I was looking at the website and I'm definitely going to be back for other seminars because it looks like it's just a fantastic series that you have. So thank you. And also thank you to Renato for not only inviting me, but explaining to me what UTC time was and making sure I got here on the right time. So I'm going to talk today about an application of Bayesian learning. And I'm going to do it in sort of a historical way to explain how we ended up using Bayesian optimization to solve some biological problems. The problem we're going to be tackling today is actually a relatively simple one for biology. We're going to try and predict what a bacterium needs to grow. So, what do bacteria need to eat before they can grow and multiply and cause disease, as we'll see. So, we're going to start by talking about traditional, sort of non-Bayesian models when they broke down, and then we're going to incorporate some human intelligence, some artificial intelligence, and then finally ask if we really need a model whatsoever. And this is going to mirror the history of AI. By accident, when we finally finished the work, we looked back and said we did the same thing that the AI field did over a much larger time scale. So the bacterium we're talking about today only live in your mouth. So they're a group of bacteria called the streptococci. They only live on human teeth, which is remarkable because no human is born with teeth or teeth erupt at some point, and then within about 2 days, these bacteria will colonize the surface of the teeth. And there's a nice happy community of the streptococci, and it isn't a problem to have bacteria on your teeth. In fact, it's usually a good thing, unless the community gets a little out of balance. So one of the bacteria in particular, Streptococcus mutans, will try and overthrow the rest of the community. It really likes dietary sugars, and to turn that into energy, it ferments them into lactic acid. If Streptococcus mutans overgrows in your mouth, you get your teeth being entirely colonized by S mutans and the biofilm they make, which we call tartar or plaque. And then the acid becomes so strong that it actually demineralizes your teeth. It burns a hole in the side of your teeth. And we call this a cavity in the United States. So, getting a cavity is an infectious disease. It's caused by bacteria. If you didn't have these bacteria in your mouth, you could eat all the sugar in the world and you would never get a cavity. So, tooth decay is actually really interesting. Most people don't believe that's it's an infection. They think about the sugar, and sugar is connected because that's what the bacteria eat. But it's a very common disease. So almost everyone has had a cavity or dental caries, which it's called in other parts of the world. In fact, in the dental literature, about a third of us have untreated dental caries. We probably should go see a dentist and have it taken care of. It's the most common cause of surgery. So when you go in to get a cavity filled, it is an oral surgery. It's people a lot of times don't think of it because it's usually done on an outpatient basis and under local anesthetic. But some people really suffer from this. So by about retirement age, almost 30% of adults will have complete tooth loss. They won't have any teeth left. They've had to been extracted or they lose them due to tooth decay. And the impact of this disease is not evenly spread across groups. So we're not going to talk a lot about tooth decay. I just like to bring it up and highlight the importance of the disease, but we're going to talk about the bacteria that cause tooth decay um, because we don't know very much about them at all. We don't know, for instance, what they need to grow and survive. We know that they can live in our mouths, but what exactly they need to eat remains a bit of a mystery. So we're going to put the bacteria on hold for a little bit, and we're going to switch to talking about AI. All right, and in particular, we're going to wind back the clock to 1994. This was not a big moment in oral health history, but this was a big moment in AI. This is when Deep Blue, a computer, beat Gary Kasparov. So this is arguably the first time that an AI system beat a human grandmaster or a human world champion in a game. The checkers community will say that that's not true. Earlier, before this happened, a checkers program called Chinook had beat the world checkers champion. So they will claim that was the watershed moment in AI, but the chess event seemed to get much more press than the checkers world championship match. So Deep Blue worked as a rule engine. So a huge team of people, in fact, an entire academic community had tried to take the rules of chess and convert them into something that a computer could understand. So it was a rule engine. We would call this now symbolic AI, where it tried to play out through the rules and come up with good strategies and then make it smooth. It's interesting that this was actually a rematch of the that happened in 1992, where Kasparov won, and then the team Deep Blue at IBM came back a second time and won the second match. Kasparov said that Deep Blue might have cheated immediately after the match. He wasn't too pleased with being beaten and hinted that maybe there was an expert that was helping the machine along. Later on in life, he seemed to soften that view a little bit. Right. But Deep Blue was this rule engine where it has some sort of knowledge about the game of chess, and it combines that with search. So it has all these rules, it has values for different positions and different pieces on the board, and then it will search through and play ahead in the computer to try and find out what move it should do. Now, the trick to rule engines is that everyone will agree that the knowledge in the computer was inferior to the knowledge that was inside Kasparov's head. Kasparov understood the game of chess much better than Deep Blue did. But Deep Blue was faster. It could search ahead and evaluate many more positions than Kasparov could. They refer to this as sort of thinking more than Kasparov. And in 1994, we saw this inflection point where even though you had inferior knowledge, search became so much faster due to advances in computers and specialized hardware for playing chess that it finally reached the tipping point, and the extra search made up for the diminished knowledge. So when we started working on bacteria, a lot of systems biologists, including ourselves, use sort of the symbolic AI equivalent of Deep Blue to understand what it is that microbes need to survive. So our version of Deep Blue is not a computer chess agent, but actually a metabolic model of a bacteria. So this is a picture, this is a metabolic map put together by a graduate student, Keenan Jajokli, who's shown in the upper right. So Keenan went through the genome of the bacteria, particularly Streptococcus mutans, and found every gene associated with an enzyme that works in metabolism. And he assembled them all into one coherent map, one model, in essence, all of the rules that go on inside of Streptococcus mutans. And in theory, a computer can play through this map and figure out if a bacterium can make all of the things it needs to grow given a set of metabolic inputs found in the media. So each reaction is a rule, and then we simulate it using linear programming in this case, or other algorithms to make calculations. So with our rule engines, we can do lots of different things, and we can get at sort of complex questions in biology. And I want to distinguish between a couple different types of complexity that we see in biology. One reason why biological systems are so tricky is because they have all this nonlinear behavior. Uh, Now, this nonlinearity is actually not something that we're able to capture well with the type of rule engine models that Keenan built. Other biological models can handle nonlinearity better, but ours can't. They're too large, they have too few parameters. Most of the behavior is linear that comes out of them. But what our models are good at is examining complexity that arises because of connectivity. There's hundreds of enzymes and metabolites and reactions inside the model, and the sum of all of those different interconnected parts can create interesting phenomena. So here's an example. You see here in the upper left, this is raffinose, which is a carbohydrate that we wanted to know if Streptococcus mutans could grow on and how it did so. So raffinose is brought into the cell, and then there's a set of metabolic pathways that break it down into galactose. So galactose being a byproduct actually also has to be handled by the cell. So the cell now, even though it's growing on raffinose, has to deal with galactose, which is a different sugar. So the model predicted something a little counterintuitive that to grow on raffinose, you need the enzymes that you also need to grow on the sugar galactose. And it turns out that this is correct. So these are experiments that Keenan did. He deleted the genes for galactose and shows that when you delete those genes, you actually get a growth defect when it's grown on raffinose. And if you put the genes back, then the growth defect goes away. So without these galactose genes, you actually get a buildup of toxic intermediates, and it's not obvious. This galactose pathway is outside of the standard raffinose pathway. So that's an example of the complexity that you can get from the connectivity inside these biological systems. But most of what I'm going to talk about today is a different type of complexity, and that's a complexity that arises due to the combinatorial nature of biology. Okay? So there are so many moving parts that we can perturb in lots of different ways that it just explodes the number of possible environments and different states that the biological system can be in. So as an example of this, I'm going to ask maybe some of the people who are on the Zoom channel can give an answer. So there are 20 amino acids that are used by all biological systems to make proteins. And humans have essential amino acids, meaning there are amino acids that we cannot make ourselves. We have to get them from our food. The question is, how many amino acids are essential? Of the 20, how many do we need to take in? Any guesses? No, no brave volunteers on this. All right. Well, the answer is actually 11. So if you've ever read the back of food and it says it has essential amino acids, it means it refers to one of these 11 amino acids that we can't make ourselves. We can only make nine as humans. Now, the second interesting question is, well, what are the 11? And that's not a simple answer. So actually, all 16 of these possibilities will give you the essential amino acids, but they're all a little bit different. So there's combinatorics that happen in here. So sometimes if you have arginine and cysteine and glycine plus these other amino acids that will work, you can get by with just cysteine and glutamine if you have serine instead, and it goes on. There's 16 different possibilities for this, and it actually gets worse. So depending on which tissue we're talking about, different tissues, different groups of cells have different amino acid essentialities. Genotypes can matter. There are individuals who are phenylketonuria and they cannot break down phenylalanine, so they can't eat phenylalanine, in which case they have to have tyrosine instead. And it changes with age. So newborns require tyrosine as an essential amino acid. It's not essential for most humans. So it changes with location, it changes with time. This is a combinatorial explosion in these possibilities. So we wanted to answer with our model, what are the amino acid essentiality requirements for this bacterium? Which of the 20 does Streptococcus mutans need to grow? Right? So we started by doing experiments and comparing the results of the experiments with the model. So here I'm showing the 20 amino acids and whether or not we add it to the media. right? And then we started taking one amino acid out at a time. So this is an experiment where we've removed alanine from the media, and this is one down here where we removed valine. And then we have an experiment down here where we've taken all the amino acids out. Now, the model predicts that Streptococcus mutans actually doesn't need any amino acids to grow. It has all the pathways it needs to not have essential amino acids and to make them all from much simpler metabolites. But when we look at the experimental data from in the lab, that's correct for a while. So you can start to take out individual amino acids one at a time, and the model is fine. But if you take all the amino acids out, Streptococcus mutans cannot grow. It has some essentiality. It just we haven't seen it yet in our experiments. So that means the breakdown of the model, the problem is somewhere in between these leave one out experiments that we did and the leave 20 out experiment. We just need to find it. The problem is that there's over 1 million combinations between these experiments that we did. There's 21 experiments here, 20 plus the leave one in, leave all of them in, and the experiment down here. And if you're going to do this in the lab, so just doing the next set, the leave two outs, that's about 1,100 experiments once you do replicates, and that would require 23,000 different pipetting operations to be done. So simulating these in a computer, you could, in theory, run 1 million simulations with a computer, but actually getting the result, the answer from the lab, this is a Herculean task. No one was really interested in doing 23,000 pipetting steps. We weren't interested in paying for 23,000 pipette tips in order to do all of these experiments. So we decided we needed to scale this up a little bit. So we gathered a team of engineers together and said, here's what we're going to do. It's very easy to get answers to these questions from the computer. We can just say, here's a media, and then wait for the computer simulation to say, here's the growth of the bacteria. Did it grow or not? And we decided that we wanted the same sort of interface, the same level of ease when running experiments from an actual physical lab. So if I have a set of experiments that I want to run with different ingredients and whether different concentrations of those ingredients, I want to be able to just put that into a computer and get the actual result from the lab without involving any humans. So we built this system. What happens is that the user specifies a design that gets sent to a computer that converts this into a schedule for a set of laboratory robots. That schedule is used to run a couple of robots that set up source plates that contain, for example, the 20 amino acids and any other ingredients. They get sent to another set of robots that are very good at dispensing combinations into really small 384 well plates. Then we use other robots to inoculate with the bacteria or add any common reagents, and then send those to robots that are connected to incubators that will move the plates out of an environmental chamber and into a plate reader that can monitor the bacterial growth. And in the end, after all of that, and waiting about 24 to 48 hours, finally the computer gives back the growth value. This is not a prediction from the model, but it's an actual measured growth that comes from this experiment that ran inside the lab. So the to the person using the computer, it seems like you're running a simulation with a really long lag, and the data comes back, but it's not a simulation. It's actually kicking off experiments that happen inside the lab. Now, when we started right before the COVID-19 pandemic, we could do about 300 experiments a day with this system. That's individual experiments, all separate. They can be entirely different from one another. Um, we can do a couple thousand experiments per day right now, and then actually our system is going to be upgraded this summer. We'll be up to 60,000 experiments per day that our robots can handle and push through. So an amazing number of experiments that we can do using this system, but we're up against this combinatorial explosion. And if you know anything about how combinatorial growth sort of trumps any sort of linear march through experiments, you know that even 60,000 a day is not going to cut it. We thought we need to be a little smarter if we're going to navigate these huge spaces that have millions or tens of millions of possible combinations. So the first thing we decided to do is get a little clever and say, let's take a page from the combinatorial optimization book and say, can't we sort of branch and bound this space? So if you think about all these amino acids, I've shown 6 of them here, that if you take one amino acid out, that gives you 6 different possibilities. And then you can pick one of those, sort of one leaf in this tree, and then you can take another amino acid out, and that gives you another six possibilities. And this continues to branch and branch. Eventually, there'll be two to the n leaves. So for amino acids, there's about a million possible combinations, 2 to the 20th experiments that we could run. So structuring this as a tree is nice, but it doesn't really reduce the number of experiments. The trick that we use in combinatorial optimization, particularly mixed integer programming, is that we can assume a sort of metabolic convexity. So imagine if you have a particular media, I've taken out three amino acids and the bacteria stops growing. One thing you could assume is that if I took out these three amino acids and it doesn't grow in the remaining 17, taking out anything else isn't going to bring it back to life. So if it doesn't grow now, removing more things isn't going to fix the fact that it doesn't grow. That's sort of this assumption. We call it metabolic convexity. It's related to the convexity assumption when you relax in MILP. So if I can imagine that any one of these conditions don't grow, I can just stop branching the tree. And now we've reduced it quite a bit from 2 to the n down to some really small space because every cut through the tree takes off a huge branch of things that would happen below it. So that this is great. We would be able to very efficiently explore this space if this holds, but we decided we should probably check to make sure this metabolic convexity is something that applies in biology. And we got about 1 day. We did one round of experiments before we found a counterexample to this. So metabolic convexity does not hold in general in biology. Here's an example of it. So I'm showing here some growth data. They're represented as dots. The larger the circle, the more the bacteria grows. So this when you take out methionine, which is an amino acid, you actually get very reduced growth. So we get a small circle here from deleting methionine. The rest of these dots indicate what happened when you delete or take out other amino acids. And you see that this is the growth on methionine, which is really small. If you also take out the amino acid cysteine, you get much more growth. Basically, it comes back to life when you take out another amino acid. So this violates our convexity assumption. It doesn't grow without methionine, but it grows even better without methionine and cysteine. So it's not convex, and we can't use this branch and bound approach. This counterexample turns out to happen quite a bit. So this shows all pairs of amino acids. So each one of these dots is an experiment where we've taken out 2 of the 20 amino acids. And anything that's in blue here is this non-metabolic convexity. It actually grows better when you take out more amino acids than when you look at the individual amino acids taken out by themselves. Right? So all the blue here are cases where we violate metabolic convexity. So we can't use that. This branch and bound approach was not going to work in order to solve our problem. So because we didn't have sort of a way to make it through the space intelligently, while we were formulating what to do, we went with sort of a brute force approach. We just fired up the robots and let them run hundreds or thousands of experiments every day. And we did all pairs of amino acids being removed. I'm showing this for 4 different species of bacteria. Again, every square is 2 amino acids that are removed. So along this diagonal here are just the single removals. And what you should see is not any particular pattern. Just notice that the patterns are different between the 4 bacteria. These are all bacteria that live in our mouth. They all live in exactly the same spot. And yet they have completely different patterns of amino acid oxytrophies. So they require completely different sets of amino acids in order to grow. Okay? And this changes again if you grow them without oxygen. So the bacteria in our mouth actually like to grow without oxygen. That's one of the reasons they form their biofilms. And then you see again small shifts in the patterns. This is really surprising because they all have unique profiles. They all require different amino acids to grow, but they all have exactly the same metabolic pathways. So when we look in the model and try and explain these differences, we're really at a loss. According to the models, according to the genomes for these bacteria, they can make all 20 amino acids by themselves. They've just chosen not to for some reason. And each species has chosen to not make a different set of amino acids. So they're kind of like children, I like to say, that go to college and their parents always cooked for them. In theory, they could learn how to cook and survive on their own, but since they've never had to, they just chose not to. They just decided to not pick up that skill. Because we're always putting food into our mouth, the bacteria have access to amino acids all the time. And for some reason, they've lost different patterns of amino acids. So Now we're in a bit of trouble because our rule engine, our model, can't make any of these predictions. It can't get it right. And we've shown that every species has a different pattern. In fact, in different environments can change the pattern. So if we want to learn what do the bacteria in the mouth need to grow, we would have to do experiments on every strain with every possible genetic perturbation that could happen because they all have slightly different genomes. And we'd have to check in every possible media and even in every environment, right? And you can see how this combination of experiments just absolutely explodes, even far beyond what we could do at thousands of experiments every day. So I like this quote from the physicist Ernest Rutherford that there's no way we could brute force any farther into the phenotypic space. We're going to have to start thinking. So when it comes to thinking, we can actually go back to how did the AI community get out of this trap, right? Deep Blue is great, but they already started to see diminishing returns about the time that they beat Kasparov, and more search or trying to compile better rules was seeming to have diminishing returns. In fact, this is a quote from the chief engineer of the Deep Blue team who said that Go, which is another board game, is just larger. We can't brute force our way through Go because the game is bigger. There's more spaces to be played on. So he predicted that Go is not going to be solved within the next 20 years. And he made this prediction in 2002. He did it because of the combinatorial complexity. So checkers has a space of about 10 to the 20. Our amino acids was 2 to the 20, so quite a bit smaller. And checkers at 10 to the 20 was actually solved completely. This was done in about 2007. There was a science paper that solved the game of checkers. It's a draw unless one player makes a mistake. Chess has a complexity about 10 to the 46th, but Go is just astronomically larger, 10 to the 170th. So there's no way that you can use the same techniques for chess to brute force your way through Go. But although he predicted this was 20 years off, this is one of the few times where the AI community has made a prediction, then it came true early. All right, so 14 years later, we have AlphaGo come out and beat a world champion Go player. So actually ahead of schedule. So how did they do this? Well, they started with some human knowledge, like a rule engine. They downloaded all the games played by Go grandmasters, and then they augmented it with reinforcement learning and neural networks. So we try to do something similar. We have these models. They're called Cobra models. This is the one I showed you that Keenan built. The models make pretty poor predictions. So everything here, every dot here I'm showing you is one data point from our experiment. There's actually 5,200 experiments here. And if the model was accurate, all of the orange data points would be right on top of the blue data points, but you can see that they're not. We've tried things like augmenting our human knowledge model with neural networks and train them, and we get good results. We can actually improve the efficacy of the Cobra model by hooking a neural network on to help take up some of the uncertainty, to fix the things that are not in the model. We get better predictions. Now the predictions seem to match the experimental results a little better. Right. So I don't want to go into too much in depth about how we do this uh, augment our models with neural networks because I really want to talk to you about the most exciting thing that happened, which is do we need a model at all? Like, can we just skip all of the human knowledge and learn directly from scratch? And we were inspired to do this because while the AlphaGo program started with human knowledge, it was quickly defeated by another program by the same team at DeepMind that started with zero knowledge whatsoever. You could actually get better play by not biasing it with human knowledge to begin with. Okay, so we thought, can we do the same thing? Can we have an AI system that learns these metabolic requirements directly from scratch? And can it explain the findings to a human at the end? So the AlphaGo systems, these game playing systems, rely on reinforcement learning, which I believe is very closely related to Bayesian optimization, although it requires a lot more samples, usually in the RL case than the Bayesian optimization case. So in the RL loop, you have some sort of agent, typically nowadays a neural network, but you can use other type of models that design some sort of experiment. It chooses an action and then the environment responds with a reward signal, a quantitative signal that the agent can learn. And updating its predictions about this reward allows it to choose better experiments, and this runs in a loop until you get optimal performance, right? So we designed the same thing where we took our automated experimental system that I showed you, but now rather than humans choosing experiments, we said we are going to take a model and we're going to have it plan and choose experiments that then get sent to the laboratory. Right? So the growth data will come back, the model will update, it will search inside the model and choose new informative experiments, and this will run in a closed loop. And in essence, we took our entire robotic automated lab and we turned it over to the AI system and said, you get to pick all the experiments, and humans aren't going to weigh in as to what happened. So if you walk through the lab and watch the experiments happen, and they say, what's going on in the well? What are you studying? We would say, I don't know. It's actually an AI that picked all the experiments for today. It has a plan, but the humans don't. So we can actually watch the system. We call it Bacter AI. We can watch it learn over time. So every day, for this run, we allow the AI system to choose 336 experiments. That number turns out to be convenient logistically the way we do the experiments in the lab. So every circle here is one experiment that the AI chose. It's showing how many amino acids it chose to remove in that particular experiment. The model also predicts whether or not the bacterium can grow on that set of amino acids. So if it's a filled circle, it predicts that it grows. If it's an open circle, it predicts that it doesn't grow. And anything in black means that the agent was correct. Anything in red means after we get the data back from the lab, it turns out the agent's prediction was incorrect. It grew when the agent said it didn't, or vice versa. So this is day one. To start off, since we don't want to use any prior knowledge, this is a randomly initialized neural network, and we're just picking random experiments, right? And we get about half of them incorrect, which sounds plausible, right? So no knowledge whatsoever. It doesn't know these are amino acids. It doesn't know what it's trying to do. It's just purely randomly selecting experiments, and we're sending them to the lab. By day three, you can see that it's actually improved quite a bit. So it's selected different experiments. The number of red experiments, the incorrect experiments have gone down. We actually get results every day, but I'm only showing you every other day here for the sake of time. And this continues. So sometimes it isn't monotonic. It actually reverts a little bit. You see that the accuracy of the model goes down. This happens usually for a human about year 3 of your PhD, where you think you understand your system. Then you get an experimental result that comes back, and you say, "Oh, I didn't understand what's going on. I actually needed to collect more data." And you see by about day 10, the accuracy of the model is up over 90%. And by day 13, we just have it stop doing experiments, because over 90% of the time, before it runs the physical experiment, the model can already tell you what the outcome is going to be. Now, we don't let the agent repeat experiments. So the easiest way for it to score points here would be to run an experiment and then the next day choose the same experiment when it already knows the answer. It's seen the data and effort. It has to pick new experiments every day. So in essence, its accuracy is always computed over test data that's coming in from every round of experiments. So within 2 weeks now, the model has solved this problem. It can tell you ahead of time what amino acids it needs across the entire space, all million combinations of amino acids. What it's actually doing, its policy, so to speak, is that if you think about, there's one media that has all 20 amino acids, and there's one media where you take all 20 of them out. And we believe somewhere in the middle is sort of this non-convex growth front where it transitions from it grows to you take an amino acid out and it no longer grows. And it's actually learning where this growth front is. It does it in two ways. So for exploration purposes, it has just a random policy. It takes a random walk by removing one amino acid at a time until it hits the growth front where it predicts the growth front is. It also does a more sort of exploitation type thing where it tries to dive down and find the smallest media that it hasn't tested, where removing a single amino acid will cause it to transition from growth to not growth. So the classic exploration exploitation dual policy that it's using in order to collect data. Inside the agent is a neural network, and that neural network is actually learning quantitatively how much it grows. So here's again the model shown in blue. Every dot is an experimental result. If the model were perfectly accurate, all of the black dots would be right on top of the blue dots. But you see at day one, it's just making random predictions. It's predicting some intermediate growth value, but almost all of the predictions are incorrect. And you see very quickly the model learns. So now by day 13, You've seen that all of the black dots are more or less on top of the blue dots. The model's prediction is quite accurate. This uncertainty here is about the same as the uncertainty in the actual experiments. So collecting more data doesn't really improve the model. When we repeat the experiments day to day, there's small variations in the bacterial growth. So we can't really get the model to be more accurate than it is. So now the model knows how what bacteria need in order to grow. We would really like to know as scientists. We don't just want a model, but we're curious what makes these bacteria tick. So we actually made a second agent whose job is to pull the information out of the first agent and distill it down for something that's easier for humans. Okay, so this builds logical rules, highly regularized logical rules that are simple enough that a human can look at them and understand what the amino acid requirements are. So this happens asynchronously than the true data collection loop. So to compare the two results, this is what Bacter AI came up with. Our AI closed loop system said after about 2,700 experiments, it says this is what the Streptococcus mutans needs to grow. It needs isoleucine and leucine or valine. It needs one of these three, which makes sense. These are called the ILV amino acids. They are closely related. They're produced by very similar pathways. It needs glutamate or glutamine, which, as you might guess from the name, are also quite similar. And then it also needs leucine or proline, this other third. So it needs a minimum of three, one each from these three groups. Now, we had asked a graduate student in the lab who was working on metabolism, could you also come up with a rule? So they looked at 422 experiments, all the leave one ins and leave one outs and leave two ins and leave two outs to be collected. And they looked at the data and they came up with this rule that you need one of the ILV amino acids and you need glutamate or glutamine. So you notice that there's a difference between these two rules, right? There's this third clause in the Bacter-AI rule. And the question was, well, who is correct? All right, so I'll give you a moment. You can make your prediction. Was the graduate student correct or was the AI system correct? They can't both be correct. Well, if you guess the graduate student, if you take these 422 experiments and compare both rules, then the graduate student is more correct, right? This leucine and proline thing doesn't really show up. However, just before you think that the graduate student has won out, if you look at all of the experiments, so all the data that we've collected, then the AI system is correct. What actually happens is that this last clause, the dependency on leucine or proline, doesn't show up unless you've taken out a lot of other amino acids. Right? So it's not that the graduate student missed this oxytrophy, it's that this doesn't actually appear in the smaller data set. Right? So the question is not what's the best way to distill the information out of the data set. The graduate student is very good at that. Is they couldn't handle the data on the scale that Bacter AI could. And we offered to let them review all 2,700 experiments and redo their rule, but that's not something that the human was interested in doing. So the accuracy of the model depends on what you're using as your gold standard. And when you use the larger data set, the AI system wins. So we've continued to do this. We found all sorts of interesting tricks to speed up this learning process. We were pleasantly surprised that it took a couple 10 days or so to learn. We thought it would take months in order for it to figure out these complexities for growth. A couple of things work really well. One of them is transfer learning. So rather than starting from scratch, we did an experiment where we started with a model that was trained on the wrong bacterium. So after we learn one bacterium, for instance, Streptococcus sanguinis, that took about 7 days and 2,300 experiments. If we, instead of starting from scratch to learn the second bacteria, we actually started with the model for Strep sanguinis, even though the predictions were incorrect, starting on just the first day with that model cut the training time almost in half. So it only took 4 days and about 1,300 experiments to get a very accurate model for Streptococcus gordonii. So knowledge can be transferred between these species, and it works especially well because while they are different, they are still closely related species. We can also do this across environments. So if we start by just looking at amino acids, there's 20 of them. Again, it takes about 7 days to learn that. If we wanted to learn all of the other ingredients in the media, so these bacteria also need sugars, they need minerals, they need vitamins, they need cofactors, all sorts of things besides amino acids in order to grow. So there ends up being 39 actual ingredients in the defined media that we use. If you start with the 20 amino acid data, you can transfer that in, and it only takes about another week, 7 days of experiments to almost double the number of inputs. So even though the combinatorial space expands by a factor of 500,000, you only need a linear increase in the number of rounds of experiments when you reuse the data. So overall, there's sort of these three approaches that we have to predicting whether or not bacteria can grow. We started with sort of these rule engines, then we combined the rule engines with the neural nets, and now we just said, can we learn directly from the data? Just have a black box model, choose its own experiments and learn. And what we're really doing is we're decreasing our dependency on prior knowledge. So that goes down quite a bit, but the amount of data, the number of experiments we need goes up. So when Keenan built his original model, the model is quite accurate on a number of things, and he used relatively a small amount of data to do it. Maybe the results of a few hundred experiments in order to tune his model and get decent accuracy. Where really for the model free learning, we're talking the order of thousands of experiments, but we don't need any prior knowledge. So there's a trade-off between these two. And what type of model you want to build, well, it really depends on how much data you have and how much prior knowledge you want to use and your confidence in. Now, the last thing I want to talk about is sort of, well, we're talking to a general audience that interested in Bayesian decision making and data collection, is that I think a big issue that we have is that when we train data scientists, when we train students on machine learning, we tell them all sorts of tricks to improve the performance of their model and to make their model better. But the one thing we don't usually talk to them about is acquiring more data, right? That if you get more data, you can improve your models quite substantially. I think that for every field of scientific machine learning, you need three things in order to make your good predictions. You need a flexible model, something that's powerful enough to handle the thing you're going to model. You need to be able to train that model that requires compute, and you need data to train the model. And all three of these have sort of a return on investment curve where there's diminishing returns that happen. And the question is for every subfield, where are you along this curve for all three of these ingredients? Now, in my subfield, so working on microbiology, trying to predict growth, we are not compute bound. We don't have massive models that need huge GPU clusters in order to train, right? So we don't really get returns by increasing our compute power. Similarly, our models are generally powerful enough. We use just a simple feed forward multi-layer perceptron for the work I showed you today. I think transformers are that we're exploring some of those now. They seem to be powerful enough to learn microbial metabolism. What we're missing is the data. We do not have data for most things in microbiology. All right, we are entirely data bound as a field. We actually quantified this. This is a paper that came out of our group where we downloaded every publication from PubMed. And then we asked what species of bacteria is studied by each one of those papers. So there's about 43,000 species of bacteria. Um, so after we download all these papers, we found that half of all of the papers that have ever been published just talk about 12 species, about 10 species of bacteria. So E. coli is number one. It's by far the most studied bacterium. You can see that up here. But really, after you get out more than 10 species, you have less than half of the literature. And three-quarters of the species that we know about, we have their genome, we've placed them in the tree of life. There's never been a scientific paper published on them. So there is just no data available to use as prior knowledge for training a model. Even if you have a language model that can, in theory, read every paper that's been published, there's nothing for it to read. And this is actually getting worse. So we're finding more and more species all the time. The microbiome revolution is in full swing, and we are identifying all these bacteria that matter to our environment and to our health. And even though the number of papers is increasing to outpace inflation, when you divide the number of papers published every year by the number of species, that ratio is going down. So we're learning less and less on a per species basis. And the biggest issue, which I think was pointed out well by this qualitative paper that came out of Google, is that there are very few incentives to work on producing more data. Everyone wants to work on models, and there's career incentives to improve new models, new architectures, new training techniques, but not to produce large data sets, right? So this is our existential problem, is that our models can do these wonderful things if they have data, but we don't have data for most of the things that we want to study. And I think this could lead to the second or the third AI winter now, is we're going to run out of data. So in conclusion, there's a lot of corners of biology that we just don't have any data at all. But once we collect the data, whether we collect it by a human or we collect it by an AI system guided in some sort of Bayesian loop, it's actually relatively easy. If you have the right data for the type of work we're doing, simple models seem to suffice. When you're collecting data, though, you should maximize the amount of information you gain per experiment. And this is something I don't think biologists are very good at. We focus on either one at a time experiments, or we try and figure out how many experiments we can do for a fixed amount of money, and we don't really look at the information content. So I think these are the issues. They are strong opinions that are weakly held, and I'd be glad in the Q&As to hear other people's responses to the role of data in biology. I want to thank all of the people in our lab that have done this work and developed the robot, a number of funding sources that have generously contributed to this work. I have on the slide a new collaboration we're starting with Align to Innovate, where we're going to be producing a huge amount of data. We're going to take 1,000 microbes and grow them in 1,000 different environments to try and solve this problem. So stay tuned for that. There's going to be a huge data release from our lab coming in the next couple of years that I think will hopefully fix a couple of these data sparsity issues. And with that, I'm happy to take any questions, and thank you again for the invitation to talk.
Yeah, thank you so much. I'm gonna conclude now by thanking the speaker. It was a great talk. And we'll move on to the question and answer discussion period. Um, so we'll first have a short period of question and answers that'll continue to be live streamed. And then after that, we'll stop the live stream, so anyone who prefers not to be reported can go ahead and ask their questions at that point. But for now, go ahead and type any questions that you have into the Zoom chat, and then Professor Jensen can read them and answer them as we go. Since there are no questions there just yet, to kick things off, I was wondering if you could tell us a little bit more about the actual neural network that's being used here. Like, how big of a model are you needing to get that good of performance, in predicting what a
Uh,
good, oh yeah.
So the model is about as simple as you can imagine. So for the amino acid example, we have 20 amino acids that are the inputs, and we have growth, which is just a scalar as an output. We have anywhere between 3 to 5 hidden layers with no more than 256 sort of hidden nodes in between there. It's a straight feed forward neural network, nothing fancy at all in there, like you'd find in a general textbook. Um,
That's
we
pretty cool. Yeah, that's something that's simple is able to do that well at this task.
right. So
Yeah.
we originally thought we were going to use a Gaussian process regressor in order to do this, especially in the first couple of rounds, because we thought we might not have enough data to actually train and get good accuracy out of a neural network. So we did that experiment where we said, well, we're going to start with a GPR, and then we're also going to train a neural network. And as soon as the accuracy of the neural network improves beyond the GPR, then we're going to switch to the neural network. So we'll basically use the Gaussian process to start the system up in the low data regime, and then we'll switch over. So we got that all set up, and then we found after day one, the neural network outperformed the Gaussian process. So we actually never
Yeah.
used the trained GP. 300 points was enough for the small neural network that we had. So all of that code went to the wayside. It was never actually used in production. So
Yeah, I gotcha. Uh, yeah, I guess we can give it a minute or two, see if anyone else has. Oh, there we go. Uh,
yeah. So the question: have you tried using active learning approaches to handle the data bottleneck issue in your domain? Uh, I think absolutely that this is going to be the key. I think there's two parts of this. There's one is the algorithmic side to say what's the best curriculum, what's the best active learning strategy that we can use. And then the other side is how do you actually carry out those experiments? And I would say that 85 to 90% uh, of our problems, and that's just a made up statistic, just a guess, 85 to 95% is entirely logistical issues inside the lab. And we have a lot of people in the group right now just working on how can we do these experiments, get the data to be reproducible enough. So I think that eventually we're going to be in a situation where we can focus and say, yes, here's interesting tweaks we can make to the active learning. But even now, just doing the simplest thing that we can imagine, since we're always limited on the data collection side, that's where we're at. That doesn't mean that we shouldn't be squeezing more out. And obviously, we can be much more efficient if we have a better policy for selecting experiments and learning. But if you're choosing what's the low hanging fruit to go after, pushing on the lab and the logistics side has been more fruitful for us for right now. And eventually that will switch. And then I think we'll be back diving deeper into the theory. That said, if anyone is a theorist and wants to work on these issues and wants to see, put these things into practice, we'd love to talk to people that have cool ideas about how you can do experiments online. The advantage we have is that the experiments are relatively cheap compared to a lot of biological experiments. So we can do them. We've done about 1.5 million experiments with the robots out of the lab so far. Like I said, we can do thousands every day. So it's a good test bed for trying out new policies, new acquisition functions.
Cool. Okay. Well, thank you so much again. And we're now going to conclude the live stream portion of things. So before we end the live stream, I want to thank all of our listeners on YouTube for joining us. And then again, remind everyone that we'll have another seminar coming up next Monday at the same time. And again, you can check out the schedule on our website, which is gp-seminar-series.github.io. So please feel free to join us again next week. Thanks. And then we'll just give us a minute here to turn the live stream off.